question 5, it shows you figure 16, which is the structure of a hemoglobin. Okay, so basically hemoglobin is a pigment that is found in red blood cells. Okay, so one red blood cells can contain thousands, okay, hundreds of thousands of hemoglobin, where each hemoglobin will carry or will bind to oxygen molecule. Okay, so one hemoglobin will bind to 4 molecules of oxygen. So why 4 molecules of oxygen? Because in the structure of hemoglobin, which consists of protein, it consists of 4 subunits. So 1, 2, 3, 4. Okay, 4 subunit, which is 2 beta, okay, 2 beta polypeptide chain. Okay, and this one is also beta. Okay, and also the other 2 is 2 alpha polypeptide chain. So in each of these chain we'll have a hem group. Okay, so we'll have a hem group, where within this hem group there is an iron atom that functions to bind to an oxygen molecule. So for question number 1, it says here, describe the structure of hemoglobin. So hemoglobin, as you can see, it consists of 4 polypeptide chain to form a functional hemoglobin. So therefore hemoglobin, the structure of it is quaternary structure. Okay, quaternary structure. That is for the first point. For the second point, quaternary structure. And then for the second point, it consists of 4 polypeptide chains, which is 2 alpha and 2 beta polypeptide chains. And then the third point, because it has, every polypeptide chain has a hem group, so hemoglobin is considered as conjugated protein. Okay, conjugated protein because it has other substance apart from protein, apart from amino acid in the component of hemoglobin. Okay, so it is a conjugated protein. It has other non-protein component. Okay, so hemoglobin, when it binds to oxygen molecules, so it is known as oxyhemoglobin. Okay, so for question 2, figure 17 shows structure of a dipeptide. Okay, so a dipeptide consists of 2 amino acids that is joined together by peptide bond. So label the peptide bond. So we have looked at this question earlier, a similar question. So this is the peptide bond, so you have to properly label the peptide bond is between carbon and nitrogen atom within the molecule. Okay, so the next question B, figure 18 shows structure of glucose and fructose that are isomer to each other. So glucose and fructose, they are hexose, 6 carbon sugar. Okay, so, they are actually isomers of each other, where the molecular formula is the same, but the structural formula is different. Okay, where at carbon number 1, okay, where the position actually, okay, the position of the carbonyl group is different. Okay, the position of carbonyl group is different, where for glucose, the position of carbonyl group is located at carbon number 1, and as for fructose, the position of carbonyl group, this is supposed to be a double bond there, okay, is located at carbon number 2. Okay, so therefore for glucose, it is considered as aldose. Okay, aldose. And as for ketose, it is a type of ketose. Okay, so identify and describe the type of isomer for both molecules. Okay, so therefore, because they are structurally different, but both have same molecular formula, so therefore they are structural isomers. Okay, structural isomer. So 1 mark. Okay, identify, 1 mark, and then describe. You have to describe. So this the description is that like I've mentioned, they have identical molecular formula. okay, but the arrangement of atoms within the molecule is different. Okay, different in the terms of the arrangement of the atoms within the molecule is different. So therefore they are considered as structural isomer. The arrangement of atoms is different. Okay, same molecular formula, but the arrangement of atoms is different. Okay, number 2. Name the disaccharide formed when a glucose and a fructose bind. Okay, a disaccharide consists of 2 monosaccharides that are joined together. In this case, the monomers are glucose and fructose, so the answer will be sucrose. Okay, and the bond between these 2 monosaccharides, glucose and fructose, is known as 1,2 glycosidic linkage. Okay, because it forms between carbon number 1 of glucose and carbon number 2 of fructose. Okay, in the structure of sucrose. Okay, number 3, draw a structure of a disaccharide when 2 molecules of glucose bind. Label the linkage formed. Okay, so in this case, you have to draw a molecule of disaccharide that consists of 2 molecules of glucose. So you know that a disaccharide that consists of 2 molecules of glucose will be maltose. Okay, so in the question, it gives you the structure of glucose, so you have to convert it into a ring structure. Okay, so remember, I've always mentioned to you again and again, you have to know the structure of glucose. So glucose is a 6 carbon sugar. So in this case, okay, the oxygen, okay, and then you have the structure of the 5 carbon ring. Okay, okay, so and then another glucose. Okay, because maltose consists of glucose, it is of type alpha glucose. Okay, alpha glucose. So at carbon number 1, okay, this is carbon number 2, carbon number 3, number 4, number 5, and number 6. Okay, okay, so basically I'm just gonna show you, okay, the number of the carbon number 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, and 6. Okay, so now you have to label, okay, all the atoms within this molecule. Okay, so because it is alpha glucose, so the OH is at the below the plane. Okay, so same goes to this one. Okay, so OH, so you remember it like this. This is how I remember. So OH is bawah bawah, atas bawah. Okay, bawah bawah, atas bawah. Okay, so remember the carbon is bonded directly to oxygen. Okay, do not draw it like this. Okay, this one is wrong. The carbon is bonded to the oxygen atom, not the hydrogen atoms. Okay, okay, so back to the drawing. Okay, so same goes to the second glucose, bawah bawah, atas bawah. Okay, so the rest will be hydrogen atoms. So hydrogen, hydrogen, hydrogen. Okay, hydrogen, 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 and hydrogen. So at carbon number 6 is CH2OH. CH2OH. Okay, so you have to draw the disaccharide, right? Okay, so there's a bond formed between carbon number 1, okay, and carbon number 4. So you just delete out. Okay, you just remove this. Okay, because in the reaction it involves the removal of a water molecule. So OH is removed, H is removed, so now you are left with oxygen. Okay, so now the oxygen, you just erase it for a bit. Okay, this one also. Okay, so now the drawing will be, okay, the bond hydrogen is maintained with the carbon. Okay, now you draw the oxygen that left tadi. Okay, and then the carbon is bonded to the oxygen, carbon bonded to the oxygen. So now the bond is formed between carbon number 1 and carbon number 4. So the bond form is 1,4 glycosidic bond, or glycosidic linkage is the same. Okay, so this is the bond that exists in maltose. So to be specific, it is actually alpha 1,4 glycosidic bond because it involves alpha glucose. Okay, so so that is 2 marks lah. Again, so 1 mark for the correct structure of maltose. The second mark is for the labeling of the glycosidic linkage correctly. Okay, number C. Okay, question C. So figure 19 shows structure of a lipid. Okay, so lipid, again, we have looked at the concept in the previous question. Okay, so lipid in this case is a type of triglyceride because it consists of a glycerol molecule and 3 fat. No, this is not a glycerol. This is actually a phospholipid. Okay, a phospholipid molecule. Okay, how do you know? Because there is presence of a phosphate group and a choline group here. Okay, so and it has 2 fatty acid, so it has 2 hydrocarbon tail. Okay, it has 2 hydrocarbon tail. So this region that has the phosphate group, okay, or here you have a negative sign, and the choline group has a positive sign because this region has charges, so this become the hydrophilic head. Okay, so the fatty acid here will become the hydrophobic tail. Okay, so that is hydrophobic. This region does not attract water. Hydrophilic head attracts the water molecule. Okay, number 1, figure 19. In figure 19, identify glycerol and draw a box around it. Okay, so really easy. So glycerol is basically this region only. Okay, so the oxygen is from the glycerol. Okay, so this is glycerol. So you draw a box. If the question asks you to draw a box, you draw a box. You do not circle it. Please avoid that. Okay, or you just you don't point, you don't do it like this, for example. Okay, so if you just label the glycerol like this, okay, your answer would be wrong because you are not answering according to the question, where the question wants you to draw a box. So you draw a box. Okay, the next question, number 2, name the bond linking glycerol and fatty acid. So this is obviously it involves the bond between the carboxyl, okay, carboxyl and also hydroxyl here. So the bond is ester bond. Okay, ester bond or ester linkage. Ester bond or ester linkage. Number 3, identify the type of lipid. So already answered that question, which is phospholipid. Okay, and then differentiate fatty acid J and K. Okay, so if you look at the structure of J and K carefully, you notice that, okay, for the structure of fatty acid J, it is a straight molecule. Okay, so a straight molecule of fatty acid is actually a type of saturated fatty acid. Okay, so because in the structure of saturated fatty acid, all the bonds between the carbon, okay, consists of only one single bond. Okay, one single bond between the carbon, between the carbon's atom within the hydrocarbon structure here. Okay, so each of the carbon will have a maximum of 2 hydrogen bonds to each of the carbon. Okay, so it is saturated with carbon atoms. Okay, so therefore the molecule of saturated fatty acid will be straight. Okay, will be straight. Okay, will be straight. And as for the second type of fatty acid, just to note, if you wrote your answer, instead of writing saturated fatty acid, you wrote saturated fat. Okay, so your answer would be wrong because saturated fat is different. Saturated fat is triglyceride. In this case, we are looking at phospholipid. The component of phospholipid consists of glycerol, phosphate, and fatty acid. Okay, so please take note. Fatty acid and fat is different. Fatty acid is the component within phospholipid or triglyceride. Okay. Okay, so for the second type of fatty acid here, as you can see, the structure of the fatty acid is bent. Okay, so why is it bent? Because here the carbon atoms are joined together by double bond. Okay, by double bond, so that's mean each of these carbon is bonded to only one hydrogen atom. So where you have a double bond, it causes the molecule of fatty acid to be bent. Okay, or we can call it as kink. Okay, so that those are the differences between the fatty acid. So which is for differentiate fatty acid K. So fatty acid K is saturated fatty acid, whereas K is unsaturated fatty acid. Unsaturated, saturated. Okay. The second difference is that you can write, okay, for the saturated fatty acid, it has no double bond, and in case of fatty acid K, it has double bond present. Okay, double bond present. So write your answer properly. And then next, the next difference is that for fatty acid J, you can write, okay, number okay, because it is saturated. Okay, saturated, all the carbon is already bonded to maximum number of hydrogen atom. Okay, so you can write has maximum number of hydrogen atom. Has maximum number of hydrogen atoms. Okay, and as for fatty acid K, one or more hydrogen atom can be added to the carbon. Okay, one or more hydrogen atoms can be added to the carbons in the structure of fatty acid. Okay, so those are the differences that you can write for the difference between J and K. Okay, question D. Okay, figure 20 shows structure of a deoxyribose. Okay, so there's again for deoxyribose at carbon number 2, the carbon number 1, number 2, number 3, number 4, number 5. Okay, so deoxyribose, it is a type of 5 carbon sugar. Okay, so you always have to remember at carbon number 3 there is a presence of hydroxy group that involves in the reaction of condensation that will join the nucleotides together, okay, to form polynucleotide. Okay, so at carbon number 2 you have hydrogen atom. So if this were the structure, okay, if this diagram is a structure of a ribose sugar, so is for carbon number 2 it will have a hydroxyl group. Okay, if this structure is a ribose, so at carbon number 2 you have hydroxyl. Okay, so give the type of nucleic acid made from this deoxyribose. So the answer would be DNA. Okay, DNA molecule. Okay, so DNA or deoxyribonucleic acid. Okay, number 2, describe the structure of nucleic acid. Okay, so describe this the structure of nucleic acid found in D1 in this answer. So you have to basically describe the structure of DNA. Okay, so basically DNA, the question worth 4 marks. Okay, so basically to answer this question is not too difficult, where the structure of DNA will be, it will have double strand. Okay, double stranded or consists of 2 polynucleotide chain. So that is for the first mark. Okay, first mark you mention the DNA molecule consists of double stranded or consists of 2 polynucleotide chain. Okay, so for the second mark, okay, so in order to hold the 2 strands together, the 2 polynucleotide strands together, obviously there is a type of bond to join these two strands together. So as you know, the strand protruding from the backbone, you have the bases. So the bases can be adenine that pairs with thymine, guanine that pairs with cytosine, and so on. Okay, so this pairing between the bases, okay, causes the formation of hydrogen bonds. So 2 hydrogen bond, okay, between adenine and thymine, okay, guanine and cytosine, 3 hydrogen bonds. Okay, so the presence of hydrogen bonds will hold together the 2 strands of the polynucleotides, okay, the 2 strands of the DNA molecule. Okay, so that is for the second point, okay, the presence of hydrogen bonds. Okay, the hydrogen bonds hold together the 2 DNA strand, okay, between the complementary base pairs. Okay, number 3. Okay, point number 3, for DNA, the bases consist of what? Adenine, thymine, guanine, and cytosine. Okay, so you can write the symbol or you can spell out, okay, the spelling for each of the bases. You spell out adenine, thymine, guanine, or cytosine, or you can just write the symbol as ATCG, okay, whichever is accepted. Okay, so the next one is you mentioned what are the monomers for this for DNA. The monomers of DNA will be nucleotide, okay, nucleotide that are linked together, okay, by phosphodiester bonds. Okay, the nucleotides are linked together by phosphodiester bond. Okay, so those are the 4 marks that you can write to describe the structure of DNA molecule. Okay, number 3, state the location where nucleic acid in D can be found. Okay, so where can you find DNA? So DNA in your eukaryotic cell, obviously they are found in the nucleus. Okay, and as for prokaryotic cell, prokaryote, you have to remember examples of this organism can be bacteria, okay, unicellular organism that is really simple. So because the organism is really simple, they don't have membrane bound organelle. Nucleus is a membrane bound organelle. So prokaryote does not have any nucleus. They do not have any membrane bound organelle. So where is the location of the DNA, the genetic material for this organism? They are located in the cytoplasm. Okay, in the cytoplasm. Okay, cytoplasm, or to be specific, in the nucleoid region. Okay, nucleoid region. Okay, so total mark for question number 5 is 22 marks. Okay, so question 6, okay, it gives you figure 21. Okay, figure 21 shows water molecule forming hydration shell around the ions of copper sulfate. Okay, copper sulfate. So copper sulfate, so it is between copper ion Cu2 plus and SO4 2 negative. Okay, so it is an ionic interaction there. Okay, so this is the solute. Okay, so in order for solute to be dissolved in a solvent, in this case water, okay, so water molecule will surround the ions, okay, that has charges, or molecules that are polar, and this causes the molecule to, this causes the solute to dissolve, okay, in a solvent that is consists of water. Okay, so water molecule will surround the ions, okay, anions or cations or any polar molecule, to cause the solute to dissolve in water. Okay, so so the water molecules will surround these ions. Okay, so as you know, water molecule consists of oxygen, hydrogen, and another hydrogen. So the oxygen region will have a negative partial charge, and as for the hydrogen region, have the partial positive charge. So for the Cu2 plus, okay, because it has a charge of 2 plus, okay, it has a positive charge, the water molecule at the oxygen region will be attracted to the Cu2 plus, okay, copper Cu2 plus. Okay, so because it has a negative charge, so water molecule will surround the cation, kan? Okay. Okay. So this is what we call it as hydration shell. Okay, where water molecules surround the solute, or in this case ion. So we call it as hydration shell. Okay. So for SO4 negative sulfate, okay, because it has a negative charge, for water molecule, the hydrogen region, okay, the hydrogen region of the water molecule will face this ion, okay, because the hydrogen ion has a partial positive charge. Okay, so until the water molecules surround the anion SO4 negative. Okay, so to form the hydration shell. So for P, as you can see, it is the hydrogen region of the water molecule. So the hydrogen region will surround SO4 negative. So I identify P is SO4 negative. Okay, 2 negative. And as for R is the, as you can see, the region of water molecule that surround R is the oxygen region. Okay, so R will be Cu2 plus. Okay, so that is your answer. Okay, number 2, water strider is an insect, okay, that can glide and hop on the surface of water without sinking. Explain how the properties of water enable it to do this. Okay, so as you know, water molecules, okay, so this is, for example, the surface of water. Okay, so water molecule at the surface here, at the surface of water here, will form or interact with each other through the formation of hydrogen bond. Okay, hydrogen bond. And water molecule at the surface will also form hydrogen bond with the water molecule below. Okay, okay, like that. Okay, so this creates a phenomena, we call it as surface tension. Okay, surface tension is created due to the interaction between water molecule through the formation of hydrogen bonds. Okay, so because it involves many hydrogen bonds, okay, formed between the water molecule, so the interaction is quite strong, that can allow insect, okay, for example, this is a spider that has 4 legs, okay, can walk, okay, on the surface of the water. Okay, so explain how this property of water enables it to do this. Okay, so and then another point that you could write is that the surface tension is due to the cohesion. Okay, cohesion behavior of water, cohesion. Okay, so 2 marks, cohesion behavior. Okay, cohesion behavior between water molecules, and then that creates, okay, that causes the water to have a high surface tension. For the second mark, okay, water has high surface tension. You can add some more mark to explain in detail. Due to hydrogen bond that forms between the water molecule. Okay, and also the water molecules also form hydrogen bond with water molecule below. okay, below the surface. Okay, so those are the points that you can include in your answer. Okay, so the question only worth 2 marks, so enough for you just to mention cohesion and high surface tension. Okay, cohesion, high surface tension. Okay, and then due to hydrogen bonds between the water molecule at the surface and hydrogen bonds between water molecule at the surface and below. Okay, okay, so question B, figure 22, okay, shows next question B, figure 22 shows molecular formula. Figure 22 shows molecular structure of an amino acid. Okay, so for question 1, draw the Zwitter ionic form of an amino acid. Okay, so Zwitter ionic form is formed when the amino acid is ionized in aqueous solution. Okay, so here, okay, where you have the carboxyl group, here, the hydrogen will be released, okay, into the surrounding solution, and the surrounding solution, if it's has so many hydrogen ions, so the nitrogen or the amino acid can accept another hydrogen ion. Okay, so where you have released the hydrogen ion, so you have a negative sign at the oxygen, so the overall drawing will be something like this. Okay, so the nitrogen will have 3 hydrogen, okay, a plus sign at the nitrogen atom, nitrogen bonded to the carbon side chain, hydrogen, oxygen, carbon bonded to the oxygen, and oxygen that has a negative charge. Okay, so that is Zwitter ionic form, where the amino acid has both positive and negative charge. Okay, so so the question worth 2 mark. Okay, so 1 mark for the correct structure of the amino acid, 1 mark is for the correct placement of the charges. Okay, number 2, amino acid are amphoteric molecule. Explain the importance of amphoteric properties. So amphoteric molecules is basically a molecule that has both amino group and carboxyl group. Okay, so any molecule that has both of these has both acidic and basic properties. Okay, they can be both acid and base at the same time. Okay, at the same time. Okay, so what explain the importance of this property? Okay, so for the first point, okay, you can mention has both acidic and basic group. Okay, has both acidic and basic group, or for the same point you can mention amino group is ionized into NH3 plus and acidic group is ionized into COO negative. Okay, point number 2 is that when an amino acid can ionize, okay, in this form, they have negative and positive charge, so by releasing and accepting hydrogen ions. Okay, so therefore amino acid can function as pH buffer. Okay, they can control the buffer of the solution. Okay, so because the amino acid function as pH buffer, okay, another point that you can include in your answer is that the amino acid can, the presence of amino acid in certain solvent, okay, or medium, it can resist the changes in the pH of that solution, okay, or maintain the pH of certain solution. Okay, maintain the pH or resist the changes in pH. For example, your blood, okay, your body fluid, the pH of your body fluid or your blood is maintained at 7.4. Okay, how to maintain this pH? Your body fluid will have so many different protein that function as pH buffer to make sure that your body fluid or blood is maintained at 7.4. Okay, for example, the hemoglobin. Okay, okay, number 3, give an example of a defense protein. Okay, protein that are present in your blood, for example, to protect from infection of pathogen that can cause disease. Okay, so you can have antibodies. Antibodies, or another term for antibodies, is immunoglobulin or Ig. Okay, or you can write lysozyme. Okay, lysozyme, which is a hydrolytic enzyme found in cells, which is to hydrolyze. For example, okay, this is a macrophage. Okay, macrophage that engulf pathogen, for example. So as the pathogen is engulfed within the macrophage, okay, it forms a vacuole that fuse with lysosome that contains lysozyme that will later will break down the pathogen, okay, inside the macrophage. Macrophage is a type of immune cell. Okay, immune cell. And then, okay, so those are examples of defense protein. Okay, see figure 23. Okay, it says here, figure 23 shows molecular structure of a disaccharide known as malt sugar. Okay, so as you can see, this is basically the structure of maltose. Okay, carbon number 1, number 2, number 3, number 4, number 5, okay, number 6. Number 1, number 2, number 3, number 4, number 5, number 6. Number 6. Okay, so the bond form is between carbon number 1 and number 4, that forms 1,4 glycosidic linkage, or alpha 1,4 glycosidic linkage, because it consists of the monomers are alpha glucose. Okay, so is this a reducing sugar? Explain your answer. Okay, so you know that for disaccharide there are 3 types of sugar: maltose, okay, maltose, lactose, and fructose. Sorry, sucrose. Maltose, lactose, and sucrose. Okay, of these 3, only 2 are considered as reducing sugar. What does it mean by reducing sugar? It reduces the copper ion, copper 2 plus, to become copper Cu plus. Okay, from Cu2 plus, okay, jadi Cu plus. Okay, from copper 2 sulfate, it become copper oxide, which is when you heat this solution that contains a Benedict reagent with your food sample. So you heat the solution. Okay, if the mixture changes color from blue to brick red, for example, it indicates that your sample contains reducing sugar. So in this case, why it says is maltose is a reducing sugar? Yes, it is a reducing sugar. So 2 marks. So for the first point, is your answer will be yes. Yes, you get 1 mark. Why? Because like I've mentioned, if your food sample contains reducing sugar, it changes the blue color. Okay, it changes the blue color of Benedict reagent. Okay, from blue to brick red. Okay, from blue to brick red, precipitate, or other range of color. It can be, the color can change from blue to green, or from blue to orange. Okay, so the different color, green, yellow, orange, brown, brick red, indicates that your sample contains different concentration of sugar. If green, contains low concentration of reducing sugar. If brick red, it contains the highest concentration of reducing sugar. Okay, so if your sample contains reducing sugar, it changes the blue color of Benedict solution from blue to brick red range, or another range, blue, yellow, green, orange, okay, or brown. And another point that you can add is that yang tadilah yang saya mention, the sugar will reduce copper to ion Cu2 plus to Cu plus. Okay, copper ion. Okay, copper Cu2 plus to copper oxide Cu plus. Okay. So those are the answer that you can include in your answer lah. Okay, so so next question, number 2. Suggest what happens when the sugar is hydrolyzed. So hydrolyze is basically a reaction that breaks any polymer or disaccharide back into its monomer. Okay, so in this case, maltose is broken down into its monomer, which is glucose. So hydrolyze, from the word lysis, to break. And the reaction requires a water molecule. Okay, so suggest what happened when the sugar is hydrolyzed. So you can write, okay, the glycosidic bond is broken for 1 mark, or the product, you can write the product which produces 2 glucose molecules or 2 monosaccharide that separates into 2 monosaccharides. Okay, so next question 3, compare the structure of amylose and glycogen. So amylose is one of the component of starch. Okay, so starch has 2 components actually, either amylose or amylopectin. Okay, so amylose is the simpler form where the structure of amylose is only coiled. Okay, coiled structure. And as for the amylopectin, you have branches. Okay, short branches. And as for glycogen, the structure of glycogen is similar to amylopectin, except for the structure is much more complex where it has more branches. Okay, so glycogen are storage carbohydrate that are found in animals, and then the glycogen are stored in liver. Okay, starch is the storage carbohydrate that are found in plant. So compare the structure of glycogen. Okay, so the question wants you to give your answer in terms of structure only. Okay, structure, do not give anything else. Okay, you don't mention in terms of function. For example, similarity between amylose and glycogen is that they function as storage carbohydrate, so your answer would be wrong. Okay, so that is in the term of function. So your answer should describe in terms of structure. So the similarity of both amylose and glycogen is that they are composed of alpha glucose monomer. Okay, alpha glucose monomers, or both contains glycosidic bonds, or both are polymers of carbohydrates. Okay, so those are the similarity of these 2 molecules. Okay, and the difference is that for amylose, okay, it is unbranched or linear. And as for glycogen, the structure of glycogen is highly branched. Okay, and then another difference is that you can write in amylose there's only the presence of 1,4 glycosidic linkage, and as for glycogen, additional to 1,4, you have 1,6. Okay, 1,6 glycosidic linkage that exists between the branches. Okay, where you have branch, there's 1,6 glycosidic linkage. Okay, so 2 marks lah, kan? So 2 marks, 1 mark for similarity, 1 mark for the correct differences, okay, between these 2 items. Okay, glycogen and also amylose. Okay, D1, complete the table below to show the differences between triglyceride and phospholipid. Okay, triglyceride in terms of structure. Okay, you have to give in terms of structure. It already gives you for triglyceride, which is a glycerol that has 3 fatty acid. For phospholipid, it has 1 glycerol. Okay, 1 glycerol, 2 fatty acid, and 1 phosphate group. Okay, so write your answer properly. Okay, and then for function, as you can see, for phospholipid, it is the major component of cell membrane. And as for triglyceride, the function of it is to is that it functions as energy storage or source of energy, or function as thermal insulation. Kan, thermal insulation macam kita tengok soalan yang lepas, animals that live in really cold environment, okay, such as whale and walruses, they have a thick layer of fat. We call it as blubber. Thermal insulation. Okay, you can write your answer for triglyceride as thermal insulation. Okay, for animals that live in really cold environment. Okay, number 2, explain how hydrogenation changes the property of vegetable oils. Okay, so vegetable oil will be liquid at room temperature due to the presence of double bond between the carbon that causes the triglyceride molecule to be bent. Okay, to be specific, the fatty acid component is bent. Okay, so this prevents the molecule from closely packed together. So in order to change vegetable oil, okay, to margarine, for example, which is in solid state, so the vegetable oil has to undergo hydrogenation. Okay, hydrogenation. So where you have the double bond between the carbon, so you break the double bond, so there's only one covalent bond between the carbon. Okay, because you want to add, you want to maximize the number of hydrogen atom bonded to the carbon. Okay, bonded to the carbons in the hydrocarbon chain. Okay, so therefore for your answer, explain how hydrogenation changes, okay, the properties of the vegetable oil. For the first point, you can write the process, okay, convert unsaturated fat, so this is unsaturated fat, okay, into saturated fat, okay, from vegetable oil to margarine, for example. Okay, the process convert unsaturated fat into saturated fats. Or, okay, so level of saturated fat increases and the level of unsaturated fat decreases. Okay, so from liquid become solid, so therefore the amount of saturated fat will increase as compared to unsaturated fat will decrease. Okay, and and then for the second point, you can add hydrogenation. Obviously you add more, okay, more hydrogen atoms. Okay, adding more hydrogen atom to the carbon that has previously the double bond. Okay, so next, the next point that you can write is that cis double bond. So this is previously this is a type of cis double bond. Okay, cis double bond where the hydrogen is on the same side. Okay, cis double bonds can convert into trans double bond. So from cis convert to trans. Okay, like that. Okay, trans. So trans double bond. So cis fat to trans fat to become more solid. Okay, so those are the answer that you can write for your answer. Okay. Okay, so next, figure 24 shows structure of DNA. Okay, question F, the last question. Figure 24 shows structure of DNA. Okay, so DNA consists of double-stranded polynucleotide. Okay, the first strand and the second strand. Okay, so as you know, phosphate is of one nucleotide is bonded to the to carbon number 5. Okay, carbon number 5 here. Okay, and so carbon number 5. So this will be oxygen. carbon number 1, number 2, number 3, number 4, number 5. Okay, the branch is carbon number 5 that is bonded to phosphate. And then you have the next nucleotide, and then the next nucleotide, and then the next nucleotide along the chain. Okay, so the last nucleotide of the chain here, okay, you have the 3' carbon that is bonded to oxygen, hydroxyl group. Okay, so if you were to add more, okay, more nucleotide to the pre-existing strand, okay, nucleotides to the pre-existing strand, you always add the new nucleotide at the carbon that is of the sugar that is labeled as number 3 here. Okay, we call it as the 3' end. Okay, 3' end. So you have the 5' end here. Okay, and the 3' end. So always add new nucleotide through the reaction of condensation, okay, to the existing strand. You always add the carbon number 3 of the last nucleotide. Same goes to this strand. So this is phosphate, carbon number 5, okay, carbon number 5, number 4, number 3, number 2, number 1, and the oxygen. So the last, so this is one nucleotide, okay. This is the next nucleotide, and then the next nucleotide along the chain. So this is carbon, this is oxygen, carbon number 1, number 2, number 3, number 4, number 5. So as you can see, carbon number 3 has hydroxyl. So if you were to add more nucleotide to the existing chain, you always add the new nucleotide at the 3' end, at the sugar that has carbon number 3, because at carbon number 3 you have a hydroxyl that involved in the process of condensation, that the process involved the removal of water molecule. Okay, if you were to add more nucleotide to the chain. Okay, so that's why for DNA strand, okay, for example here, you always see that the DNA strand is labeled as 5', and then the other strand is labeled as 3'. So one strand, okay, and this strand will end with 3. This is 5. Okay, so if you were to draw it like a straight chain here, okay, so 5, 3, 5 has to pair with 3 complement. Okay, 3 has to pair with 5. Okay, so as actually the direction, so this is what we call it as the directionality, okay, of the strand. So the direction will go from, okay, let me just change the color, okay, from 5 to 3, okay, 5 to 3. So why is it 5 to 3? Because it begins with 5' and ends with 3'. Okay, for the 5 carbon, you cannot add any more reaction because it already bonded to phosphate. Okay, the one that involved in the reaction is carbon number 3 of the last nucleotide. Okay, you always add at carbon number 3 of the last nucleotide in order to elongate the strand. Okay, so in the question it says draw a box. Okay, draw a box along the backbone structure. Okay, so the backbone will, is the repeating unit of the polynucleotide. So repeating unit will be phosphate, okay, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. The same goes to this one, phosphate, sugar. Okay, phosphate, sugar, all the way. So, okay, so the backbone. Okay, so the backbone will is this one. So the question asks you to draw a box. So this is the backbone. Okay, the backbone. So simple question there. So repeating unit of phosphate sugar along the backbone of DNA or RNA consists of repeating unit of sugar and phosphate. Okay, so next, the last question it asks you, DNA polynucleotides are anti-parallel. Okay, anti-parallel. Okay, are anti-parallel to one another. On figure 24, write the orientation for both strands. So saya dah draw tadi kan. So the orientation will be anti-parallel kan. So anti-parallel where the first strand is going from 5 to 3. The second strand is going from 5 to 3. So this is what we call it as anti-parallel. parallel. So the question asks you to write the orientation. So yang arrow tadi lah kan. So arrow dia sentiasa 5 ke 3. Okay, 5 ke 3. Alright. So that's the end of the discussion for chapter 1. So I hope you understand. I hope you watched all the videos discussing from structure from objective until the end of question 6. Okay, thank you.